This is a production of Cornell University.
Thanks, Gary, for the very generous introduction, and thanks everyone for being here today. I'm excited to talk to you about my dissertation research on Fusarium graminearum. There we go. All right, so I'll start with a refresher on Fusarium graminearum biology for everyone. This is a fungal pathogen of wheat, corn, and other grasses. It reduces crop yields and contaminates grain with mycotoxin. And economically, it causes direct losses of about $1 billion each year in the United States. It also produces very striking red pigments in culture. It can be identified on host plants by this characteristic pink salmon sporulation, and infected grain is visibly pale and shriveled compared to healthy grain, as you can see here. This is a very simplified life cycle, but I need you to remember it for the rest of my talk. The pathogen overwinters in dead plant tissue, and in the spring it produces airborne ascospores. Fusarium graminearum is homothallic, so a single individual is able to produce these sexual spores, and they can potentially travel several kilometers before landing on and infecting host plants while they're flowering and particularly vulnerable to the pathogen. Roots and stems are also susceptible to infection through the soil or debris at the soil surface, and this can lead to additional stem colonization and pathogen overwintering. The picture on the left is of parathecia, which contain ascospores on a corn stalk, and corn stubble is generally considered the primary source of disease-causing inoculum in agricultural settings. The other picture here is of a wheatgrass panicle with a single infected spikelet. The bleaching symptom is typical, and if you look at the right side of the spike, you can see the anthers poking out, indicating that infection occurred early during the flowering process. This pathogen is extremely well studied. We have successful breeding programs. We use effective fungicides on over 70% of planted acres. We have weather-based models to predict risk in real time, and we have these promising biological control agents being developed. But we still see 10% losses on the whole, and at the individual field level, severe disease epidemics can result in a complete loss. So a big picture question might be, what else could we be studying about this pathogen? And you can probably guess my answer based on the title of the presentation. It's wild hosts. Wild hosts are interesting for a number of reasons. First of all, most of what we know about Fusarium graminearum was discovered in an agricultural context, really focusing on wheat and corn. But Fusarium graminearum doesn't exist solely in cropping systems, and in fact, cultivated crops are only a fraction of the pathogen's host range. It can infect well over 100 species spread across more than 24 plant families. And I want this slide to give you a sense of that scale. Each circle here represents a plant family, and their relative size indicates how many hosts are contained within each. All of these hosts could potentially influence disease epidemiology or pathogen evolution, but the research I'm sharing today focuses on hosts in the Poaceae, which is the true grass family. It contains the largest group of host species, as well as the most important agricultural crops. Other reasons to focus on grasses include their widespread distribution and remarkably high acreage. These are satellite images showing wheat and grass ground cover in New York and Kansas. And I've chosen Kansas as an example because it's the largest wheat producing state in the country, with 6 to 7 million acres planted a year, but grass covers 3 times that land area. And I like this image especially because it shows that interface between the wheat and the grasses. In New York, though, the disparity is much greater at almost 20 to 1. And earlier I mentioned corn being an important part of the disease cycle. And for reference, New York still has twice as much grass as corn, making this an especially good place to study Fusarium in these isolated grass communities. And in addition to the sheer numbers of these hosts, they're often growing in close proximity to crops. For example, fallow fields, pastures, or even in the margins of wheat fields themselves. And that presents a lot of opportunities for the pathogen to move back and forth between hosts. So despite their widespread distribution, very little is known about the interaction of Fusarium graminearum with wild grasses. And the goal of my dissertation research has been to characterize the pathogen's biology and wild grass hosts at a population and community level, and then to relate this new information back to our understanding of disease in wheat fields. We've been looking at this Fusarium grass relationship from a few different perspectives, and I've divided my talk into 5 sections based on the topics listed at the top of the slide. And it starts with a study on the incidence of Fusarium graminearum in wild grasses. And I put this photo here because the grasses I'm talking about are almost always infected asymptomatically. And this was the only time that I saw Fusarium growing on the outside of a grass spike. And if you look closely, you can see the sporodocia. They're bright orange and they contain the asexual conidia. So a little more background, like I just mentioned, these asymptomatic associations are well documented in surveys from around the world. but no other study has considered the environmental factors that might influence pathogen distribution. We wanted to answer these two questions: is incidence homogeneous across the state, and what factors might contribute to grass colonization? To do that, we conducted a field survey of grass spikes and overwintered grass stems from very different locations. We collected samples from 23 sites over 3 years. We focused on 12 common grass species, and we also collected infected wheat spikes from crop fields. The sites were divided between two regions of New York, the central region, which contains most of the state's grain production, and the northeast, which has virtually no wheat or other small grains and much less corn. So there were samples from regions with high and low agricultural production. We also sampled locations under two different land uses, one being agricultural. So these are wheat fields where I collected grasses from around the margins. The other land use was natural, and this included remote regions in the Adirondacks, the Montezuma National Wildlife Refuge just north of Cayuga Lake, and other non-agricultural environments that had undisturbed grasses growing. So in total, we collected more than 3,000 grass spikes and 3,000 stems. We isolated Fusarium from this tissue, kept count of the incidence of graminearum, but also held on to all of the other Fusarium species, and that comes into play a little bit later. So we compared Fusarium and graminearum recovery rates between region and land use. Plots here have region on the x-axis, percent pathogen incidence on the y-axis, with stems in the plot on the left and spikes in the plot on the right. Land use is color-coded. In both cases, there was less Fusarium in the northeastern part of the state. That difference was very clear looking at the infestation rates of overwintered stems, but the variation in spike incidence was much larger. And while on average there are fewer infections in the northeast, the difference is less obvious. And that's because a lot of the variation is tied up in the weather that we experienced during each of our sampling years. So here's the same data, split by year instead of region and land use. 2015 was an epidemic year for Fusarium head blight, and at one sampling site, all of the grass spikes collected contained the pathogen. And that high level of infection provides a lot of opportunities for the pathogen to colonize stem tissue and then overwinter, which is why we see a high number of stems infested the following year in 2016. There's a severe drought in 2016, which meant few spike infections that season, and in turn that results in less overwintering in stems the following year in 2017. We wanted to move beyond those categorical differences, so we modeled pathogen incidence and spikes based on two continuous variables, rainfall and host density. I put these graphic representations here for you to see how much variation there is across the state. Cumulative rainfall in May and June was chosen as a proxy for relative humidity, which is known to impact spore production, release, and crop infection. And our rainfall estimates were taken from the PRISM climate group at Oregon State. The plot on the right shows host density in the year prior to sampling, which is an indirect estimate of pathogen inoculum production in the year samples were collected. And the image here shows the total acreage of grass, corn, and small grains, including wheat, at 1 kilometer intervals. And this makes for a simple visual, but in reality, we use a slightly more complicated variable. And in our model, host acreage was summed within 10 kilometers of every sampling site, and acres were weighted based on their distance from the sampling site. So hosts 10 kilometers away were considered much less important than hosts within 1 kilometer where we collected a sample. And we settled on this 10-kilometer combined acreage after testing the hosts together and separately across a range of distances from 1 to 10 kilometers. So the output from our model of grass spike incidence suggests that both density and rainfall are required for grass infection, which makes good biological sense, and this corresponds to higher inoculum production as well as the environmental conditions that would be conducive to disease. The plots on this slide show 95% confidence intervals around predicted pathogen incidence rates. X-axis is your cumulative rainfall estimate, and the y-axis is the percent of predicted spike infections. And as you move across the slide from the left to the right, each plot has an increasing number of host acres. So the leftmost plots resemble what we saw in northeastern New York, and the plots on the right are more similar to what we would have seen in central New York. And so you get this rapid rise in pathogen incidence with increasing rainfall once there's a certain level of background host density. Another trend we noticed in the data was a tendency for Fusarium graminearum to displace other Fusarium species at field sites with more rainfall and higher host densities. And this is a plot using the same model as before, but now including the ratio of Fusarium graminearum to other Fusarium species rather than just graminearum incidence. And at low host densities, it's roughly a 50-50 split between graminearum and other species. But when conditions favor the spread of graminearum, we see that ratio change pretty drastically. And this is probably because graminearum produces those aerially dispersed ascospores and none of the other grass-associated Fusarium species are known to do this. So higher rainfall would especially benefit graminearum over other Fusaria. To wrap up that section, grass debris is a likely source of disease-inciting inoculum and could increase the risk of epidemics in nearby crops. Incidence in grass spikes varies with annual weather patterns and host density in a manner consistent with what we know about crop hosts. And there was an unexpected finding where we saw this change in Fusarium species communities associated with those same factors. So the next part of the talk focuses on how exactly those communities differed and why this could impact agricultural crops. Again, a little more background. As I've mentioned, multiple Fusaria can inhabit grass spikes. Many of them are also crop pathogens, and most of them produce some cocktail of toxins. So we wanted to find out what other Fusarium species were showing up in our grass samples. In addition to that, I wanted to figure out how the Fusarium communities might be structured. So we sequenced gene fragments from about 400 unknown isolates, and we identified 10 different Fusarium species in addition to graminearum. Some of them were well-known crop pathogens, several were common saprophytes, but there were two unusual finds that I think are fun to point out. This is the second time Fusarium camptoceras has been found in the US, and typically it's seen on rotting tropical fruits like banana and cacao. And this is the only time Fusarium lactis has been associated with a grass host. It's mostly known for infecting sweet peppers, but it also causes an internal rot of figs when it's introduced to the fruit by fig wasps, like this one pictured here. So there were certainly some interesting species to be found. And when we compare species diversity across sites, we can see the diversity was slightly lower in central New York than in northeastern New York. And the plot here just shows Simpson's diversity index, where a lower number means less species diversity. Another way to compare species diversity between locations is to look at differences between the communities taken from different field sites. And when we do that, we see a significant shift between the communities from our two regions. Each point on this plot represents a field site, and the distance between points shows the relative difference between their Fusarium communities. The color scheme has changed now. Orange represents northeastern New York, blue central New York, darker colors stand for the agricultural sites, and lighter for the natural sites. And you can see that while the regions are slightly separated, the two land uses are not differentiated at all. We wanted to see what was actually different between these communities, so we ran a principal components analysis on our species abundance data, and it turned out that 98% of community variation could be explained by just two species, graminearum and spore trichoides. And this is not too surprising because if you just look at the raw frequency of all 11 species isolated, shown here in the pie chart, these two are clearly the dominant members. What's more interesting, though, is that two distinct Fusarium communities were associated with each of these dominant species. Graminearum was generally found alongside less common species and saprophytes, while spore trichoides was associated with other well-known crop pathogens. This heat map here shows the strength of co-occurrences between species, and I've added boxes around the two clusters to make the divide between those communities more clear. And this is important because the various species in each of these groups can produce a different variety of toxins. So if you were trying to predict what contaminants might appear in harvested grain, understanding the environmental factors shaping these communities could be a useful predictive tool. And then just to recap that, surveying wild grasses revealed some interesting Fusarium species diversity, and understanding how communities form under those different conditions could be valuable for predicting disease risk. After looking at species and community diversity, we took a look at the genetic diversity within Fusarium graminearum populations. And to put this into context, we need to start from a very broad point of view. Fusarium graminearum is really a species complex with over a dozen member species. These member species are more or less restricted by geography, and this neat little map just shows their suspected geographic origins. But the most important member retains the name Fusarium graminearum, and it's globally distributed. It's also by far the most important species in North America. Looking at just the United States, Fusarium graminearum in that narrow sense is the predominant species. There are 3 populations that exist in overlapping geographic ranges, shown here on this map of the US. And these populations are roughly defined by their production of different mycotoxin variants, also known as chemotypes. And these chemotypes can be predicted from single gene markers. I don't want to get bogged down in their chemical structures, so I've just listed the 3 most important chemotypes and their associated North American populations here. While the populations do overlap, isolates of each are present in different ratios that change across north-south and east-west gradients. The North American 1 population is predominant in the south and west, but as you move north or east, you find more of the North American 2 population. This NA3 population was only discovered recently, and its known geographic range is much more restricted. The prevailing hypothesis is that this chemotype evolved after the others and originated in the extreme northeastern United States or in Canada. And I point all of this out because New York sits conveniently at the intersection of these gradients. Because of this, New York has a very unique mixture of these 3 populations, and we also happen to have the highest known frequency of these NX2 producing isolates that corresponds to that NA3 population. Based on previous work in Gary's lab, looking at isolates collected from wheat, corn, and airborne spores, we know that the 3-ADON and NX2 producing isolates occur at a higher frequency only in the northeastern part of the state. And these pie charts represent that. So the most important parts of background information for this part of the talk, there are 3 distinct populations that are mostly differentiated by their chemotype. New York is a particularly interesting place to study these populations. Isolates derived from wild grasses have not been considered in previous population genetic studies. And as a reminder, we collected wheat isolates during our field survey. So based on that, there are a lot of questions worth asking, but these are the 4 that I'm going to tell you about. Do land use or wild hosts influence chemotype ratios? How well do neutral genetic markers compare to our single locus chemotype markers when comparing the populations? What amount of gene flow is expected, and by extension, how much pathogen movement could occur between hosts? And do different hosts or sites serve as reservoirs of genetic diversity? To start with, we estimated chemotype frequency using 900 isolates, and then we compared their occurrence based on different factors. We did not see a difference between the 3 host sources, wheat spikes, grass spikes, or grass stems. And there was a significant change in chemotype frequency between central and northeastern New York as expected. But there was also a significant difference between land use at sites in central New York, where the prevalence of the 3-ADON chemotype was expanded in natural settings compared to what we found in agricultural fields. And we would consider that to be a novel and pretty interesting finding because it suggests that even at relatively small geographic scales, some unknown factor is able to maintain different ratios of these populations across sites. Building on that analysis, we used 8 microsatellite markers to genotype these same isolates, and these neutral markers do correspond pretty closely to the chemotype assignments. The scatter plot is from a discriminant analysis of principal components, which I've used here to show how well prior group assignments compare to observed genetic structure. Each point represents an individual isolate. They're color-coded here by population, and the discriminant analysis is attempting to separate the populations as much as possible. And in this case, the plot shows that while the genetic distance between our groups is small, they can be clearly differentiated based on the chemotype labels. The inset at the bottom left of the plot is just there to indicate that we've captured about 90% of the variation in our data set. And the density plot in the top right of the slide shows the values for just one of the axes in the scatter plot. And I want that there to drive home the overlap between these predefined populations. You can also numerically quantify population differentiation between groups, and there are 101 ways to do this. I just want to share one common measurement with you. The table at the bottom right of the slide shows a pairwise fixation index, in this case Hedrick's GST, which ranges from 0 to 1, with 1 being complete differentiation and 0 being no differentiation whatsoever. Looking at the pairwise values here, the NX2 chemotype is less differentiated than the 3 and 15, supporting the pattern of population overlap shown by the discriminant analysis. And generally speaking, those numbers would be considered to indicate moderate differentiation. Running the analysis a second time, but setting population groups based on region and land use, we see much less differentiation. Though there are some bumps in the data showing that subsets of these groups are more distantly related than others. You can see, for example, this more dispersed group of isolates from the natural sites in central New York, marked in light blue. And I think the density plot also shows this pretty well, where you can see these second peaks for the natural sites in both central New York and northeastern New York. Looking at the fixation indexes for these groupings, there's a slight differentiation between all groups, but much less than we saw between chemotypes. Finally, here's one more look at this data where groups were defined based on host source, right? Wheat, grass spikes, grass stems. And because there's virtually no differentiation here, you might infer that unrestricted gene flow and by extension, possibly pathogen movement could occur between all 3 of these hosts. Last thing I want to mention about this relates to genetic diversity and how it was evenly spread between isolate sources. So of the 700 isolates that we fully genotyped, only about 50 were identified as clones, which means genotypic diversity was extremely high, as in 0.99 on a scale of 0 to 1. We also saw equivalent allele diversity, listed here as mean allele numbers, and gene diversity, and even the number of unique alleles from each source was more or less equivalent. So to recap that, while grasses are a reservoir of neutral gene diversity in one sense, they don't contain a higher level of diversity than wheat does. Pathogen movement between these hosts does not appear limited by any kind of population structure. Non-agricultural environments support a different ratio of pathogen chemotypes than wheat fields. And because we suspect genetic recombination is occurring between 3 populations with different toxin production profiles, we should be on the lookout for novel toxin production or altered virulence in these populations, particularly at sites where we might find high admixture rates. So we saw very little genetic differentiation between isolates taken from grasses and wheat, suggesting no major constraints to colonization of different host spikes. And to follow up on this, we examined some of the other interactions between the pathogen and individual grass hosts. A little more background, host pathogen interactions in this Fusarium grass system have not been characterized very well, and Fusarium graminearum can also cause root and crown rots. The central question we wanted to get at here was how competent are various grass hosts? We measured the host competency of 5 grasses during 3 stages in the pathogen life cycle, and to be clear, I'm defining host competency as the ability to support pathogen survival, reproduction, and infection. So we examined each of these in separate experiments. The first was a winter survival assay that went on for 2 years and measured pathogen persistence in individual stem pieces that were artificially inoculated and left outside. This bar chart shows pathogen recovery from the stems of various wild grasses and wheat, Triticum aestivum, which for the next couple of slides is always going to be shown on the far right of the x-axis. The black bars here are pathogen survival rates after a single year, and the gray bars show survival after 2 years. 4 of the 5 grasses tested were comparable to wheat. One host was significantly worse as an overwintering site than all of the others, and this was clearly related to how quickly the stems had degraded. And there was also a steep decline in pathogen survival between the first and second year, which agrees with past research that focused on wheat stems alone. The second experiment was a spore production assay conducted under controlled conditions. These are 95% confidence interval plots of spore production on different host stems. The y-axis is log transformed, and spore production was adjusted to dry grams of host tissue before comparison. We also averaged the spore production of 10 diverse isolates on each host. So one grass was less suitable as a substrate for spore production than the others, but all of them supported the production of huge numbers of both ascospores, shown on the plot on the left, and conidia, the plot on the right. And again, you can see wheat is comparable to most of these grasses. And finally, the third experiment was a seedling root rot assay, where we recorded the number of seedlings infected after inoculating seeds at germination. We used 5 different isolates for this, and they were chosen because they have these different chemotypes, and I've split them apart here because there was a significant interaction between host and isolate, meaning that some of the isolates tested were more or less virulent on different grasses. And there were 2 grasses that seemed pretty resistant to infection, 3 others that were all susceptible but showed lower incidence rates than wheat. This indicates that at least some level of host specialization could exist in different isolates, and clearly the pathogen is better adapted to infecting wheat than grass seedlings. To wrap that up, all the hosts were suitable substrates for spore production. Most of them were comparable to wheat as overwintering sites, and 2 different grasses were not susceptible to seedling root rot. And together, those results suggest that various grass hosts could have different selective pressures for the pathogen. To build on that last point, we wanted to compare the fitness of some isolates derived from wheat and grass to see if there was any separation in phenotypes that might be related to their host of origin. There's no more background here because you're all experts on this by now, and the question is straightforward enough: does fitness differ between host sources? We took a dozen isolates from wheat and a dozen isolates from grass spikes, all collected at the same field site, and we phenotyped them in 9 different experiments where we measured growth, reproduction on different substrates, and virulence on 2 crops. This radar chart displays the mean relative fitness of isolates from both sources, and each spoke on the wheel just marks a different experiment. So right away, you might be able to see where some of the differences could be. Wheat-derived isolates displayed by the gray area were better at producing conidia or asexual spores and had a slight advantage when infecting wheat or corn. Grass-derived isolates, shown in blue, were better at reproducing asexually, generating more parathecia on 3 of the 4 substrates we tested, and they also had marginally faster growth rates. Based on those rough comparisons, we formally tested for differences in mean phenotype values of interest, and we found that wheat-derived isolates produced larger conidia and more conidia than grass-derived isolates, as shown here in these 2 plots. The finding just highlights the importance of looking at phenotype distributions in addition to genotypes when studying populations, and it also suggests that wheat and grasses could be selecting for different traits relevant to agriculture. This is worth investigating further because grasses could actually serve as a buffer against the evolution of traits detrimental to agriculture. For example, providing refuge from fungicide applications and helping to maintain low levels of resistance. And I think I can sum most of what I've talked about today to these 2 points. 1, wild grasses are suitable hosts and likely sources of inoculum based on surveys, experiments, and inferences from population genetics. And 2, the factors that were strongly influencing pathogen incidence, communities, and population structure generally operated on 10-kilometer or greater scales. And I think both of those ideas are relevant to land use choices, like where to plant crops, and could be incorporated into disease risk assessments. Gary alluded to the fact that I have quite a few hobby projects, and I'm not going to get into all of them today. I just want to give a shout out to some of the great collaborators I've been able to work with at Cornell. Matt Ryan's lab is doing some very exciting work on cropping systems that incorporate perennial grains, including an intermediate wheatgrass called Kernza, which is pictured here on the left. And I've been able to team up with Eugene Law to hunt for pathogens in their field trials, and it's been a real treat to look for disease in these new hosts. I've also worked a lot with Mark Sorrell's plant breeding program. Scout in the Fields has grouped plants all over the state, and has provided a lot of data on disease incidence patterns, as well as great information on crop variety performance. And some of these field sites are also in pretty interesting places, and I took this photo of Jay and Alyssa at the Wegmans Organic Research Farm. And with that, I want to thank Gary for having me in his program. It's been an incredible experience, and I think it's fair to say that he has given me the freedom to pursue my interests wherever they lead. And a few of you might even remember my first student seminar when I talked about trying to identify pitcher plant diseases, which is pretty far removed from field crops and really a testament to how much freedom I was given to explore here. I also need to mention some of the undergraduates I've worked with, and the students on this slide were all involved in my dissertation research. Kelly and Julian, who I unfortunately don't have pictures of, helped me conduct some of the laboratory experiments. Eniola was characterizing Fusarium communities last summer. She had never worked in a lab before, but did a really fantastic job, and now she's looking out for her next research experience. Mencius Kwan extracted more DNA than anyone should ever be asked to, and now he's in a PhD program at Purdue. And Jay Winans was an all-around superstar. He did an undergraduate thesis with us. He worked on every part of this research, and he also helped with 3 or 4 other projects. Right now he's working with Nancy Keller at Wisconsin-Madison. Obviously, there are a lot of other people who have helped me, and I hope I haven't forgotten any of them. So thanks for listening, and I know I have plenty of time left for questions. So ask away.
Questions? I have a question that I want to see if my interpretation makes sense based on Fusarium. So I found that result of the difference between asexual versus sexual spore production in the wheat versus wild grass really interesting. And it seems like it would be consistent with, say, the hypothesis that they're better at producing sexual spores, there's more genetic recombination, and maybe that makes them more suited to a more genetically diverse host population versus a more genetically uniform host population where there might be an advantage in producing more asexual spores that take less energy to spread more quickly. Does that make sense based on the biology of Fusarium?
Yeah, so the question is about the preference for asexual versus sexual reproduction depending on host diversity. And I think that hypothesis could make sense. Unfortunately, the data we had didn't show a significant difference between the two. And the overlap in phenotypic distributions for isolates that you collect is pretty wide. So it would be hard to get a sample size large enough, I think, to answer that definitively. But it's definitely an interesting idea. Yeah, Michael.
Well, just a follow-up to that, I guess I'm not quite sure I understood either the question or the answer just then, but graminearum is like almost completely strictly homothallic, isn't it? So how much recombination goes on?
Yeah, so the question is about how sexual Fusarium graminearum is, and we see it outcross if there's another isolate available for it to reproduce with.
From your data, can you make any inferences about that?
I probably could, but I have not yet. And that is something I could analyze using the microsatellite markers potentially.
Thanks for that really very interesting seminar, Mike. So what are the implications for plant breeders? Is there likelihood that, we don't have very good resistance in wheat, but with all that diversity, do you think that that this pathogen can overcome what little resistance we even have?
Yes, that's a great question about how much diversity is in Fusarium graminearum and what that means for breeding programs. I think one interesting thing to do would be to use a much wider range of isolates when assaying for resistance. And yes, it certainly would pose a challenge for any resistance you find, having this wide range of phenotypes in the field. But I think another potentially interesting thing to look at is the gene-for-gene interactions maybe that are happening in wild grasses in the Fusarium pathosystem, and whether any of those might give insight or hints into what kind of resistance could be useful in wheat in addition to what we're already trying to breed for. Yeah, sure, Bill.
Do you see any widespread distribution of particular genotypes of Fusarium graminearum?
So, question being about distribution of single clonal genotypes. Yeah. So we genotyped 750 cultures. We found 50 clones. Occasionally they were in the same place. Generally they were in different places. We know long-range dispersal is a potential thing that could happen. But with such a small number of clones, it's hard to say what that distance might actually look like.
Following up on Mark's question, were you able to assay any of your wildgrass samples for mycotoxin levels? Because what we're more interested in breeding is not reducing incidence, it's reducing mycotoxin. So you said a lot of the wild species are asymptomatic. Just curious if they have maybe you have mechanisms that are stopping mycotoxin production.
Yeah, so the question is about mycotoxin accumulation in wild grasses, and while we didn't look at that specifically, there is another group that has done that. And under natural conditions, the grasses accumulate very little to no mycotoxin whatsoever. If you inoculate them in a greenhouse, though, then they blow up, and sometimes you can see symptoms, and then you have accumulation of DON and other mycotoxins as well. So yeah, there's definitely something interesting there that I think would be really cool to look at more.
Any other questions? I don't think we have Geneva on today. Okay. All right. Thank you very much.
Thanks.
This has been a production of Cornell University, on the web at cornell.edu.